Today we're going to talk about what can happen if oxygen is available in this cell respiration process. So this is a mitochondria. Mitochondria have a few important structural characteristics that we want to look at. One is that they have an outer membrane. They have an inner membrane that's convoluted, lots of folds in it, so that it can increase surface area for doing this reaction. And then there's a matrix inside of the inner membrane where a lot of the processes we're going to talk about will take place. The number of mitochondria in a cell really will vary depending on the demands of the cell. If you have a cell that is constantly using energy, like your heart, you're going to have thousands of mitochondria per cell. If you have cells that rarely need energy, like fat cells, you're not going to have that many mitochondria. Your muscle cells, your skeletal muscle cells, will vary depending on the demands you put on them. So if you're a distance runner, you might have thousands of mitochondria per muscle cell, and if you're a couch potato, you might only have several hundred mitochondria per muscle cell. Your muscle cells will increase and decrease depending on the demand. That's what getting in shape. Part of getting in shape is increasing the number of mitochondria you have in each one of your cells, so that you can convert sugar to ATP more efficiently. That's what part of what getting in shape is producing more mitochondria. That's why it takes a few weeks for that to take place. So the link reaction is going to be a reaction that's going to take place after glycolysis. We have pyruvate. Pyruvate, we're going to use some energy. So really glycolysis is a wash, you might say, because the energy produced during glycolysis is used pretty much to move pyruvate into the mitochondria. So you have to go through the outer membrane and through the inner membrane into the matrix here. And when we get into the matrix, this is where we are going to do our link reaction. And what the link reaction is going to do is it's going to turn this 3-carbon molecule into a 2-carbon molecule. So we're going to lose some CO2. We're going to make a little bit of NADH. So this is the NAD is going to be reduced. It's going to gain electrons and hydrogen. And we're going to bind an enzyme to the pyruvate molecule. And it's actually an acetate molecule at this point, and we're going to make acetyl-CoA. So we have an enzyme that's going to help this molecule enter the Krebs cycle, which is the next cycle we're dealing with. So the Krebs cycle is going to take place inside the matrix, just like the link. So both these reactions, link and Krebs, take place in the matrix. And the acetyl-CoA is going to enter the Krebs cycle, and it's going to go through the pretty complicated process involving lots of enzymes and lots of reactions. None of them are listed here, but if you take biochemistry in college, you'll have to learn them all. But there's several steps to this process. One of the steps is involves citric acid. So this is actually often called the citric acid cycle as well. So Krebs was the guy who discovered the citric acid cycle. So in this process of the citric acid cycle for AP biology in high school, these are the levels that you need to know. So for every acetyl-CoA, we're going to make 2 CO2s. We're going to make 3 NADHs. We're going to make an ATP, and we're going to make an FADH2. Okay, we have 2 of these from each. Remember, for every glycolysis, every time we start breaking down sugar, we get 2 pyruvates. So there's going to be 2 of these entering. So if we talk about a single sugar molecule, you're going to end up with 6 of these, 2 of these, and 2 of these, if we're breaking down a single sugar molecule. All right, so now the electron transport system is a fairly complicated process. This is where most of the energy that we've been storing in these hydrogen carrying molecules, this is where most of that energy is going to be released. Just to give you an idea of what we're dealing with here, this membrane that you're going to see on the next page, much larger, is the inner membrane. Okay. This space right here is the matrix. That would be in the blue. And this space out here is the inner membrane space. That's the space between the outer and inner membrane. Okay, so that's what we're looking at in this next picture. All right, so this process is called oxidative phosphorylation. Oxidative because we are oxidizing all of the NADHs and FADH2s. Remember, when they lose electrons and lose their hydrogens, they're being oxidized. Okay, so we're oxidizing NADH, we're oxidizing FADH2. Phosphorylation means we are going to add a phosphate group to something, and over here, we're adding phosphate groups to ADP and making ATP. So that's our phosphorylation. So what's going on in this process? Well, the NADH is adding its high energy electrons, and its electrons are going to go through something called an electron transport chain. And in the process of going through this chain, the energy that's stored up in the electrons is going to be used to pump hydrogen out from the matrix to the inner membrane space. The electrons in NADH have the potential to pump 1, 2, 3 3 protons out. Okay, so we end up with 3 hydrogens being pumped out for every NADH, and we get 2 hydrogens pumped out for every FADH2. Okay. At the end of this process, these electrons get picked up by oxygen. That's why we call oxygen the final electron acceptor. It's also why oxygen is needed for this reaction. If you don't have oxygen, you can't pull the electrons off. You can't pull the electrons off, you can't get them to flow through. You have to have oxygen around. Oxygen molecules pick up the electrons. The oxygen molecules each become negatively charged. And when they become negatively charged, they happen to become attracted to positively charged things. And because H plus is abundant inside here, the negatively charged oxygens attract to the positively charged H plus, and they form covalent bonds, and we get water. So water is going to be one of the waste products of this reaction. And the big reason we do this whole thing is we pump all these hydrogens out. Now, the only way the hydrogens can get back in is they flow through ATP synthase. This is an enzyme that drives the production of ATP. For every hydrogen out here that flows back through, I can make 1 ATP molecule. So how do I get 34 ATP molecules? Well, it's pretty simple math. We have 10 NADHs, and each one of those is going to pump out 3 hydrogens. Okay, where do we get our 10 NADHs? We got 2 of them from glycolysis. We got 2 of them from the link reaction, and we got 6 of them from the Krebs cycle. So that's 10 NADHs. So we have 30 pumped out hydrogens, so that as those flow back in, we're going to make 30 ATPs. We had 2 FADH2s from the Krebs cycle. That's going to pump out 4, and as they flow back in, they're going to make 4 more ATPs. So here's where we get our 34 ATP molecules from oxidative phosphorylation.